This Embryology Highlight screencast, Early Embryogenesis 2, covers the development of the nervous system, a process called neurulation, somite formation and development, transition from a flat embryo to a cylindrical body, and the formation of pharyngeal arches. We begin with an image of the 3 germ layers that give rise to all of the organs of the body. This is a transection through a flat embryo. The blue shows the ectoderm, the yellow shows endoderm, brown shows the mesoderm and the notochord. Actually, the nervous system here has already developed in this flat embryo. This is the neural tube, and these are neural crest cells. They have been induced to form by the notochord. Stages of nervous system development are of neurulation. The first thing that happens is that under the influence of the notochord, ectodermal cells overlying the notochord become tall columna and form a neural plate. Neural ectoderm cells of the neural plate undergo a shape change which converts the plate into a neural groove. Shoulders of the groove come together and form a neural tube and bilaterally neural crest. Neural tube becomes the central nervous system, that is the brain and spinal cord, and the neural crest cells become neurons of the peripheral nervous system. They also become pigment cells in the skin. They become cells of the adrenal medulla, and actually they become several other tissues which we'll discuss when we get to the head. This cartoon shows the various stages of neurulation. First, under the influence of the notochord, ectoderm cells become tall columna. They can be referred to as neuroectoderm, and they form the neural plate. As these cells change their shape, the neural plate becomes a neural groove. At the junction of the neural groove with the normal ectoderm, you can detect the future neural crest cells. As the 2 shoulders of the neural tube come together, the neural crest separates, 2 shoulders meet, form a complete neural tube that separates from the overlying ectoderm. Finally, the neural crest cells segregate to form bilateral separate masses of tissue. We have the neural tube then that's going to form a central nervous system. Neural crest is going to form glial cells and neurons of the peripheral nervous system, and the ectoderm is going to form skin. This cartoon shows longitudinal views of neurulation as well as the transverse views that we have been looking at. So here we see that closure of the neural tube occurs in the region of the neck, and then proceeds rostrally and caudally. Also notice that the rostral end of the neural tube develops these enlargements, and they will then become brain, whereas the rest of the neural tube, which is simpler, will become spinal cord. This shows an actual tissue image of an embryo with the neural tube, which will become brain and spinal cord, and here bilaterally are the cells of the neural crest, which will give rise to the neurons of the peripheral nervous system and the glial cells of the peripheral nervous system, as well as non-neural tissue such as pigmented cells and various structures that we'll see in the head. This is a drawing of an embryonic brain and embryonic spinal cord, and you can see peripheral nerves, cranial nerves coming from the brain, and spinal nerves coming off the spinal cord. This cartoon shows cells migrating away from the neural crest to become neurons of autonomic ganglia. They will be located in the wall of the gut, in the abdomen, and close to the vertebrae. And some cells stay in place and they become the unipolar neurons of spinal ganglia. So both the autonomic ganglia and neurons of the spinal ganglia come from neural crest. Next, I'd like to discuss somites. This is an image of a 10 millimeter pig embryo, and you can see these chunks of mesoderm that appear just beside the neural tube. They develop first in the neck region, and then with age they appear more caudally. Somites are well developed cranially in the embryo, caudally not very well, so that this is a continuing process. You can even age an embryo by counting the number of somites. Somites are formed by the most medial mesoderm, the mesoderm that is just beside the midline axis, and so is called paraxial mesoderm. Initially, paraxial mesoderm forms one continuous mass, but under the influence of the notochord, transverse fissures appear, segmenting the mesoderm. Somite cells within the segments of mesoderm actually become epithelioid. They return to being mesenchyme again as they migrate away. Those somites that develop rostral to the notochord in the head are called somitomeres, because they are not as well developed as typical somites are. Each somite differentiates into three regions: a sclerotome, which becomes the axial skeleton, vertebrae and ribs and bones at the base of the skull; the dermatome, which gives rise to the dermis of the skin; and then finally a myotome, which becomes the skeletal muscle of the body. This shows a scanning electron micrograph that is colored. It is a transverse section through an early embryo that is still relatively flat. We can see the ectoderm, colored in blue, the neural tube, the notochord, and the endoderm, which is colored in yellow. The mesoderm is red. Laterally is where the coelom has redeveloped, is somatic and splanchnic components of the lateral mesoderm. This is intermediate mesoderm that's going to form kidney and gonad. This is the medial or paraxial mesoderm that is going to give rise to somites. This is a transverse section through an older embryo that is starting to become tubular rather than flat. It is a tissue section that has actually been colored. You can notice the ectoderm externally in blue. This is the neural tube. This is the notochord. Endoderm is down below. We have mesoderm then. This is a somatic mesoderm that's going to form body wall, separated by coelom from splanchnic mesoderm, which is going to form gut wall. And this is intermediate mesoderm, which hasn't developed into very much yet. This is a vessel, the aorta. This is the paraxial or medial mesoderm. It is colored to show the three regional parts that it develops into. This is sclerotome. This is going to form axial skeleton. These cells are going to flow as a mesenchyme surrounding the neural tube to form a vertebra. The outer region here is going to form dermis. This is a dermatome. It's going to blend with the adjacent ectoderm to form skin. This red area is the myotome. It is going to receive innervation, and once it does, these cells will migrate to mainly the body wall region, but also in the region around the vertebrae, and give rise to all of the skeletal muscles of the body. This cartoon shows an earlier embryo and a later embryo. In the earliest stage, we see the three regions of a somite distinguished by color. The sclerotome is blue, the myotome is red, and the dermatome is hatched. At a later stage, we see that the sclerotome has migrated. Those cells have formed a vertebra around the neural tube. The dermatome cells are migrating to join the ectoderm to form skin. But in red, then, we have the myotome. Having received innervation from the neural tube, those cells are going to migrate out into the body wall and form various muscles of the body. Initially, the embryo is flat, but it soon develops a cylindrical shape. And that process of going from flat to cylindrical involves three processes, if you will. The formation of a head process, the formation of a tail process, and involvement of lateral body folds enveloping the embryo and meeting at the ventral midline. On the right, we see three stages of head process development. The head is growing upward and forward. That growth is occurring in the region of what would be the future neck region. So The head is sort of growing out from the neck, and this original part that was present is going to become the cranium, and the growth is occurring in this region. In the process of forming a head, a foregut or pharynx is also formed. The embryo grows forward and grows over its anterior region, creating this foregut or pharynx. Now, the tail fold develops in a similar fashion, except that the growth is less pronounced than it is in the case of head development, but one gets a hindgut. So the foregut and the hindgut are formed in the process of forming a head process and a tail process. For most of us who are limited in our spatial imagination, it is challenging to envision how this head process forms. But you really don't have to envision the process. All you have to know is that initially the embryo is flat, and then it grows a head and a foregut in the process of differential growth. How it does that is not so critical as knowing that it's flat and then it grows a head and a foregut. Also, as the embryo grows up from its flat initial status, lateral body folds are draped to the side and then meet on the ventral midline to complete the tubular formation of the embryo. This cartoon shows 4 stages of development in the process of going from a flat embryo to a tubular one. Initially, the embryo is flat and blends with the fetal membranes, which are shown in yellow. There's some evidence of a head process and a tail process, but for the most part, the embryo is flat, blending with the fetal membrane. In step 2, we see that the embryo has grown upward above the fetal membranes, and the lateral body folds have draped to the side. Except where the yolk sac is, they are going to meet on the ventral midline and complete the tube. And 3 and 4 just make this more obvious. These are fetal membranes here, this is the yolk sac, and here the embryo is clearly separated above that. Lateral body folds have draped around. Anteriorly and posteriorly they meet on the ventral midline, completing a cylindrical body form that's obvious in this specimen here, except where the yolk sac communicates with the midgut. There, the lateral body folds are precluded from meeting on the ventral midline because the yolk sac is in the way. This is a crude drawing of an early embryo when it's still flat, and you can see that as these lateral body folds come around and meet at the ventral midline, that the splanchnopleur, composed of splanchnic mesoderm and endoderm, is going to form the gut wall. The somatopleur, composed of somatic mesoderm and ectoderm, is going to form the body wall, and they're going to be separated by a coelom. Here we see some images with the coelom colored yellow. Initially, the embryo is flat, it starts to raise up, and we have these lateral body folds starting to drape around. Initially, we don't know where the embryo ends and the fetal membranes begin, but as the cylindrical body starts to take shape, now we know that this flat part is extraembryonic and that this part here is going to be the embryo. And so that process of lateral body folds coming together and meeting toward the midline continues, and that's shown down below. Here we see on the right that splanchnic mesoderm has formed the gut wall, and it's also formed dorsal and ventral mesenteries. Meanwhile, somatopleur has formed body wall. Down below here we see that organs can develop in the mesenteries. So here's liver developing in ventral mesentery, and here's pancreas developing in the dorsal mesentery. Lateral body folds cannot meet at the midline where the yolk sac is present. And so we have the gut communicating with the yolk sac. And then eventually, as the yolk sac disappears, the lateral body folds can meet, but there is no ventral mesentery because that was part of the yolk sac and that got absorbed into the gut. Pharyngeal arches border the pharynx, which is the rostral part of the foregut. Each of these arches is demarcated by a cleft on the outside and a pharyngeal pouch on the inside. And so these two structures demarcate these various arches. Pharyngeal arches are significant because they become various things in the adult. For example, the first arch develops into upper and lower jaws, and fourth and sixth become larynx. First pharyngeal pouch becomes an auditory tube. In fish, you see more pharyngeal arches. In mammals, the first three pharyngeal arches are visible exteriorly. But in fish, pharyngeal arches, which are called branchial arches, branchial means gills, are well developed. The tissue separating them disintegrates, so there are spaces then between the branchial arches, and the branchial arches become gills. Here we see a drawing of a pig embryo, and we can distinguish three pharyngeal arches. In fact, the first pharyngeal arch is starting to develop two processes. This will be the lower jaw, and this will be the upper jaw. This crude drawing shows how pharyngeal arches are separated by clefts on the exterior and by pouches on the interior. And so you can see how this tissue could break down and give you gills. This textbook image of pharyngeal arches shows that each of the arches contains a vessel. It also contains a cranial nerve that will innervate whatever this pharyngeal arch becomes. Myotomes from somites and from semitomeres migrate to these arches to contribute the muscle to whatever structures these become. This concludes our Embryology Highlights session dealing with early embryogenesis 2.